Good afternoon and welcome. I'm George Daly, the Dean of Harvard Medical School and the chair of the Warren Alpert Foundation Prize Selection Committee. I'm very excited to be part of this wonderful event. I'm just very sorry that we're not able to join together in person, but such are the realities of the current pandemic. This afternoon, we celebrate the momentous achievements of three physician scientists, Jens Juul Holst, Joel Haebner, and Daniel Drucker. Their collective work has elucidated the function of key intestinal hormones and their effects on metabolism. These findings informed the subsequent design of treatments for type 2 diabetes, obesity, and short bowel syndrome. Their insights have reshaped the field of endocrinology and altered the treatment of cardiometabolic and gastrointestinal diseases. The scientific achievements we honor today represent a major advance against long-standing, daunting challenges in some of the most refractory chronic medical conditions, diabetes, obesity, and metabolic disease. The discoveries made by our honorees were born out of vexing scientific questions and made on a quest to alleviate human suffering. So for me, they are powerful and reassuring reminders that through the rigor of the scientific process and with the passion of researchers and physicians, we can and we will make progress in improving human health. Metabolism is an exquisitely complex hormonal symphony whose understanding flows from the work of many scientists. However, it was the key discoveries made by our three award winners that truly moved the field forward and catalyzed the design of new classes of disease-altering treatments. In the 1970s and 80s, this trio of researchers identified a family of glucagon-like peptides. Since then, they have led the field of metabolism research with studies that went from bedside observations to the lab bench and then back to the clinic. The scientists' work, encompassing research in cells, animals, and humans, led to the development of at least three classes of new medications that have profoundly changed treatments for type 2 diabetes, a disease that affects more than 30 million people in the United States and nearly 500 million people worldwide, a pandemic, if you will, in its own right. Moreover, these treatments have recently shown cardioprotective effects, a critical consideration in a population that has elevated cardiovascular risks as a result of their conditions. Yet another class of drugs informed by the scientists' work is now being used to correct absorption syndromes in people with short bowel syndrome and other conditions marked by intestinal insufficiency. What an elegant demonstration of the power of basic science to yield knowledge for the betterment of humankind. And that is, in fact, the very mission of the Warren Alpert Foundation. It is indeed because of this vision of the Warren Alpert Foundation and its founder that we are gathered today to celebrate this transformative work. Some of you have no doubt heard the origin story behind the Warren Alpert Foundation, but I will recap it for those of you who haven't. It was 1987, and Warren Alpert came across a news article that described the work of Sir Kenneth Murray, the British scientist who developed a vaccine against hepatitis B. In awe of this feat, Warren tossed the paper aside and cold called Murray to inform him that he'd won the Warren Alpert Foundation Prize. Now, Warren, of course, at the time, neglected to mention the fact that such a foundation didn't exist yet. So Warren got to work. He approached Daniel Tosteson, then Dean of Harvard Medical School, and asked him to convene a panel of experts to help choose future award winners. And the rest is history, 33 years in the making. Over 3 decades, not counting today's winners, the foundation has awarded some $5.5 million to 72 scientists, 8 of whom have gone on to receive Nobel Prizes. I'd like to thank the Warren Alpert Foundation for its stalwart support of science and discovery. Now, before we dive into the beautiful science of this year's award-winning discoveries, I would like to virtually present our honorees with the plaque. Now, of course, I really wish I could be handing this to you in person, but another sign of the times. I should note that you will be receiving an actual physical plaque in the near future. So congratulations to our distinguished award winners. And now I'd like to turn this over to our two MCs for this virtual event, both of them renowned metabolism scholars, my esteemed colleagues, Professors Ron Kahn and Jeff Flyer. Let me begin by welcoming Jeff Flyer, my admired predecessor in this deanship. Jeff.
Well, thank you, George, and good afternoon, everyone. As a member of the Alpert Award Committee and a longstanding researcher in the field of metabolism, it's a special pleasure and honor to introduce today's virtual Alpert Symposium. I'd like to begin by placing this Alpert Symposium on glucagon-like peptides in the context of the history of metabolism. Some of you may know that the first hormone to be discovered was secretin, identified in London in 1902 by Bayliss and Starling. Secretin was an extract of duodenal tissue found to stimulate pancreatic secretion after injection into dogs. Discovery of this gut hormone launched the field of endocrinology, the study of hormones and their diseases. Now, long before the discovery of secretin, von Mering and Minkowski in Germany observed in 1889 that removing a dog's pancreas produced diabetes, a discovery that stimulated many efforts around the world to find a glucose-lowering factor within the pancreas. But it wasn't until 32 years later, in 1921, that the Nobel Prize-winning discovery was made in Toronto that an extract of pancreas that was named insulin lowered blood glucose in both diabetic dogs and soon thereafter humans. That discovery profoundly altered the treatment of diabetes and catalyzed many areas of bioscience research. And the 100th anniversary of insulin's discovery will be celebrated next year in Toronto and around the world. Now, soon after insulin, a second pancreatic hormone was discovered, this time in 1923. How did that occur? While seeking to increase the efficiency of insulin extraction from the pancreas, investigators found a fraction in pancreatic extracts that raised rather than lowered blood glucose when injected into animals, and they named that molecule glucagon. Now, glucagon has important physiologic roles, but a limited therapeutic role, and has generally played second fiddle to insulin in the metabolic orchestra. Research on insulin, however, catalyzed many areas of bioscience research. Elucidation of insulin's amino acid sequence in 1952 garnered Sanger the first of his 2 Nobel Prizes. Glucagon's amino acid sequence was determined 5 years after insulin's in 1957. And then in the 1970s, recombinant DNA technology revealed the insulin's cDNA and gene sequences. And in the wake of that, in 1983, insulin became the first recombinant human therapeutic. It was in this setting, in the 1980s, that the story of glucagon-like peptides arose, most notably through the work of today's 3 award winners, Joel Habener, Daniel Drucker, and Jens Holst. As is often the case in biomedical research, these 3 scientists came to the problem looking to answer different questions, armed with different tools. What they shared was a commitment to following the science wherever it might lead. And their research uncovered novel peptides that were homologous to, but quite distinct from glucagon, named glucagon-like peptides, or GLPs, and they discovered many of their important biological activities. They found that glucagon and GLPs were processed from a larger precursor protein encoded by the glucagon gene locus, and this processing occurred in a tissue-specific manner. Their discoveries added immeasurably to our understanding of physiology and disease and enabled multiple new therapeutics that are now beginning to rival insulin in their clinical impact, as we will hear during the course of this afternoon's symposium. Now, Joel Habner, working at Mass General Hospital and HMS, was a pioneer in the use of recombinant DNA technology to elucidate peptide hormone biosynthesis and expression, initially focusing his work on parathyroid hormone, which he showed was made as a larger processed precursor. Turning his attention to the pancreatic hormone glucagon, Joel discovered in the early 1980s that the glucagon cDNA encoded a large prohormone, and he deduced that in addition to glucagon, this prohormone very likely generated additional glucagon-like peptides with then completely unknown tissue expression and function. Now, Dan Drucker joined the Habner lab as an endocrine research fellow in 1984 and began to investigate the expression of the large proglucagon cDNA in a variety of tissues. In 1986, he found that unlike beta cells that, as expected, produced glucagon, gut cells preferentially processed proglucagon to generate novel GLPs, a key discovery. One more bit of history. Prior research had suggested that the intestine might produce factors that explained why insulin release was greater when glucose was administered by mouth compared to when it was given intravenously, despite the same blood sugar levels. These hypothesized gut-derived factors that potentiated insulin release were termed incretins. Might GLP-1 be an incretin? Work conducted by Drucker in the Habner lab, published in 1987, showed that a truncated form of GLP-1 did indeed potentiate glucose-stimulated insulin biosynthesis and secretion from beta cells. At about the same time in Copenhagen, Jens Holst and colleagues were also exploring the potentiation of insulin secretion by gut factors and the biological roles of glucagon immunoreactive molecules of gut origin. Holst and colleagues demonstrated in 1986 that the proglucagon-derived peptides, GLP-1 and GLP-2, were indeed produced by the gut, and in 1987 they showed that a truncated form of GLP-1 strongly potentiated insulin secretion in vivo, an effect that waned as glucose levels fell, which turned out to be an extremely important observation. He and his colleagues described critically important activities of the hormone, including its ability to lower blood glucose after chronic administration to type 2 diabetic subjects, and its ability to reduce appetite, which much later turned out to be very important. It's important to point out that the path from these great physiologic discoveries to the creation of effective therapies, despite what you might have imagined, was neither rapid nor straightforward. There were numerous scientific, technical, and pharmacologic challenges that had to be solved, and thankfully they were. This short introduction has only touched the surface of the contributions of today's Alpert Prize winners to the exciting field of GLPs and the novel therapeutics collectively enabled by their work. You will hear more of their research and its consequence in the presentations to follow. Now, in addition to presentations by the 3 Alpert Award winners, we're delighted to welcome 2 other outstanding scientists who have long worked in this space, Fiona Gribble and Matthias Chop, and we anticipate an extremely exciting symposium. One brief note, there will be opportunity for short question and answer period after each presentation, and audience members who'd like to contribute a question can type their questions into the Q&A box on their screen. So with that, I'd like now to turn the program over to my good friend and co-moderator, Dr. Ron Kahn, who will introduce our first speaker.
Thank you, Jeff, and thanks to the Warren Alpert Committee for allowing me to co-chair this very special symposium. I want to also offer my welcome to the audience that's quite international today, listening into this symposium, so that while we have the disadvantage of not being in person for this, we have the advantage of certainly being able to have people from all over the world attend this great symposium. As Jeff pointed out in his introduction, as much as those of us who study diabetes would like to think that insulin is the sort of premier hormone, it wasn't the first hormone. And in fact, the first hormone was secretin, a gut hormone. And so we've asked our first speaker today to put this in perspective, to talk about gut endocrine hormones. And that speaker is Dr. Fiona Grable, who is a professor of endocrine physiology at the University of Cambridge and director of graduate education for the School of Clinical Medicine. She's also an honorary consultant at diabetes at the Addenbrooke Hospital. Fiona received her medical training at the Universities of Cambridge and then Oxford before gaining a PhD as an ion channel electrophysiologist. In 1999, she moved back to Cambridge and set up a joint research group with her husband, Frank Ryman, pioneering the application of single-cell technologies such as electrophysiology and live cell imaging to the study of GLP-1 secretion. This has now become part of an internationally known Institute of Metabolic Science in Cambridge. Fiona is an elected fellow of the Academy of Medical Sciences and has received a number of awards, including the Lister Research Prize, the R.D. Lawrence Lecture of the Diabetes UK, the Minkowski Prize from the European Association for the Study of Diabetes, and the Victor Moot Medal of the International Regulatory Peptide Society. Fiona's research focuses on understanding the physiology of the gut-brain-pancreas axis and how it can be targeted to develop new drugs for the treatment of diabetes and obesity. Today, she will be telling us about gut endocrine hormones as chemosensors of the postprandial state. And let me just say again before she starts that if you have any questions, please use the Q&A box during the lecture. I will collate those and we'll address as many as we can after her lecture. So with that, Fiona, the floor is yours. Thank you for coming.
Thank you very much. And it's a great pleasure and honor to join you from Cambridge in the United Kingdom this evening. It's a bright but darkening evening here. Next slide, please. So I'm going to introduce you to GLP-1 and the other gut peptides. These are peptides that come from the intestinal tract, and we've already heard that GLP-1 has major effects on insulin secretion, particularly important because its stimulatory effect on insulin secretion works in people with type 2 diabetes, hence its development for type 2 diabetes therapies. But it also turns out to have strong appetite suppressive effect. And on the left, I've shown you that it also affects gut motility, which is an interesting way of keeping the digestive system in order during the postprandial state. And at the bottom right is a picture that I'm sure will come up several times today, which is an illustration of where GLP-1 arises from in the proglucagon molecules. So you can see the proglucagon molecule contains the glucagon in pink, and then two glucagon-like peptides, GLP-1 and GLP-2. And in pancreatic alpha cells, prohormone convertase 2 cleaves out the glucagon molecule from proglucagon, and in the intestinal tract, in the L cells, it cleaves out oxyntomodulin, GLP-1 and GLP-2. Next slide, please. But as we've heard, GLP-1 is only one of a whole family of peptides that are produced in the intestinal tract. There's supposed to be at least 20 hormones produced in the gut, and quite a number of these actually influence the metabolic state. So as well as GLP-1, here I've shown you peptide YY and cholecystokinin, which are all anorexigenic peptides. I've shown you GIP, which works together with GLP-1 to enhance insulin secretion in the postprandial state. And then there are peptides that have effects on the gallbladder and exocrine pancreas. And GLP-2 down at the bottom there has a profound effect on gut health, as I'm sure we'll hear more about later. Next slide. So where do these gut peptides arise from? They come from intestinal endocrine cells, known as enteroendocrine cells, which form about 1% of the gut epithelium. So at the top right, I've shown a depiction of an intestinal crypt under the top of villus, and I've colored some enteroendocrine cells in yellow. The cells are derived actually by continuous division and differentiation of stem cells in the crypts. And as the cells migrate out of the crypts and up the villi, they differentiate into the major epithelial cell types. Most of these are the absorptive enterocytes, but there are also goblet cells producing mucus, Paneth cells producing defensins, and these enteroendocrine cells that are going to be the topic of today's talks. And they can produce a number of different peptides, as we've seen, such as GLP-1 and PYY. And at the bottom right, I've just shown you a picture of how these often look. They have an apical surface facing into the gut lumen, and then they have peptide secretory vesicles located on the basolateral side. They often have a process that is responsible for making contact with neighboring structures, such as nerve endings. And when we first saw these cells, we thought that this surface facing into the gut lumen must be there to act as a chemosensor to detect what is present in the post-digestive state. As I'll show you in the next few minutes, it's not quite as simple as that. I don't think now that this area facing the gut epithelium, facing the gut lumen, is just acting as a straightforward chemosensor. So could I have the next slide, please? So one way of mapping the intestinal tract endocrine cells is to do immunostaining, and that's the typical way that this has been done. What I'm showing you here, however, is a map that we've generated by liquid chromatography mass spectrometry. And we've taken a mouse intestine here, divided into 5 centimeter portions, and analyzed it by LCMS. And you can see on the left-hand side are hormones that are produced predominantly in the upper gut. So there's CCK and GIP and secretin. And then as you move towards the middle and to the right, there are hormones produced more in the lower GI tract. So you can see the GLP-1 and GLP-2, peptide YY and insulin-like 5. Note, next to GLP-1 and GLP-2, I have the glucagon signal. And you can see, as we've already described and discussed, that whereas GLP-1 and GLP-2 are formed in the gut epithelium, the glucagon is only produced in the pancreas. Next slide. So now I've mapped onto the, oh, sorry, could you go back one, please? Thank you. So now I've mapped onto this, the 3 boxes that actually are supposed to depict whereabouts in the gut the stimulation of these cells happens under normal physiology. So the top box is highlighting the upper gut, and this is where normal postprandial digestion and absorption occurs. And the place where absorption happens is where the endocrine secretion occurs. And you can see that it would stimulate many of the CCK producing cells are in this region, GIP producing cells and secretin. So these are hormones that go up very rapidly after you've eaten a meal and go up quite high. Then in the middle box is showing the lower small intestine. And this is the region of the gut that is stimulated following gastric bypass surgery, when nutrient absorption and digestion occurs lower down the GI tract. And bariatric surgery is very interesting in the endocrine field, because it has very profound effects on improving glucose metabolism and reducing appetite in people with diabetes and obesity. And the area of the gut it stimulates results in higher levels of GLP-1, GLP-2, peptide YY, because it's stimulating more cells further down the gut. And then the final box at the bottom there is highlighting the colon and rectum. And you can really ask yourselves why we have the enteroendocrine cells in this region. They won't be ever reached by food ingestion in a normal state, but they will make contact with any unabsorbed food components, bile acids, bacteria and bacterial metabolites. So they could be there as sensors of the interprandial and fasting state. Next slide, please. So the question that we try to address is what and how do enteroendocrine cells sense? And we know that hormonal concentrations in the bloodstream go up after food ingestion, but how does this occur? Next slide, please. And the way that we and now many other people have gone about this is to try to analyze how enteroendocrine cells work at the single cell level. So if you start this slide at the top left-hand side, you'll see a picture of glutag cells, which are a mouse GLP-1 secreting cell line. And this is where I first made contact with Dan Drucker, because he made this cell line back in the 1990s. And when we set out to try to work out how GLP-1 is secreted, Dan kindly provided us with a cell line. And we characterized actually quite a lot of function. It's an excellent cell line, and it's still actually the model cell line that is used for studying GLP-1 secretion around the world. However, our head of department at the time kept on getting onto us saying, how do you know that a cell line is going to be telling you everything that is accurate about a native L cell? They said, you really need to study the native L cells themselves. So in the Mendel panel, I'll show you that we started making a number of transgenic mouse models where we used a hormonal promoter, in this case, the proglucagon promoter, to drive a fluorescent reporter or fluorescent sensor. And we generated mice. If you look on the bottom left, this is an image of a primary culture of colonic epithelium from a mouse where the L cells are labeled by a fluorescent reporter. And you can see that most of the cells are dim, but there are two bright cells there, and those are L cells that produce GLP-1, which enabled us to do live cell and transcriptomic analysis. Now times have moved on, and if you look at the other picture on the bottom next to that, that's a picture of an intestinal organoid derived from the transgenic mouse. So intestinal organoids were developed by Hans Klever's group in Utrecht. And they allow you to take the intestinal epithelium into culture and to maintain it without, in a continuous form. You can freeze it, you can rethaw it, and you can manipulate it. And the green cells you can see in that picture are L cells that are labeled, in this case, with a FRET sensor for cyclic AMP. So you can monitor dynamic changes in cyclic AMP in the cells. And in the top right is our next iteration of this technique. Now we can label primary human L cells. So this is the first time we've been able to study human L cell physiology at the single cell level. So we've taken human ileal organoids, which we maintained in culture, and we've engineered them by CRISPR-Cas9 to express a fluorescent reporter, again, only driven by the proglucagon locus. So all those green cells, as you can see at the top right, are L cells in a human intestinal organoid. Next slide. So what can you do with them? If you look in the center bottom, this is showing that the venous, that's the yellow fluorescence, is co-localizing with staining for proglucagon, where you can see the small red stained vesicles. On the left-hand side, this shows that you can purify the cells by flow cytometry and subject them on the bottom left to RNA sequencing. So you can look at which channels, transporters are expressed in the cells. And at the top left is a peptide mass spec analysis showing high production in these cells, not only of glucagon, also of PYY and NTS, that's neurotensin. And then if you look on the right-hand side, this is functional analysis of live cells. So on the top right is an electrophysiology trace. So the cells fire action potentials. And actually, if I look at this trace, I couldn't distinguish it from a trace from a glutag cell or from a mouse L cell. You can see that action potentials are triggered when glucose is added to the medium. In the middle on the right is an example of a calcium imaging trace. This is imaged with Fura2, so you can add different reagents and see what elevates calcium. And at the bottom right is an example of GLP-1 release that you can measure in the supernatants. Next slide. So how does nutrient absorption trigger GLP-1 secretion? And L cells turned out to be a slightly unusual cell because they're embedded in the intestinal epithelium, which uses a sodium-coupled glucose transporter to absorb glucose into the body out of the intestinal lumen. The L cells seem to have hijacked the same system to act as a sensor. So SGLT1, shown on the left there, is a transporter that for every glucose molecule that enters the cell, it takes 2 sodium ions with it. And that generates a small sodium current that is sufficient to depolarize the cells, change the membrane potential, trigger action potential firing, and elevate calcium. And then the calcium is a fairly universal trigger of secretion in a variety of cell types. So in this way, the absorption of glucose through the apical membrane is acting as a direct trigger of GLP-1 secretion. Next slide. And this means that when glucose is actually, the process of active glucose absorption is accompanied by entry into the bloodstream of hormones that are being secreted in the same region as glucose is absorbed, so GLP-1 or GIP. And glucose and these hormones circulate to the pancreatic beta cells. And in the pancreatic beta cells, glucose triggers insulin secretion via KATP channels, an elevation of calcium. And the GIP and the GLP-1 act on G protein coupled receptors, which elevate cyclic AMP. And cyclic AMP enhances the ability of the pancreatic beta cell to respond to glucose. So this is how the incretin effect that we'll hear more about later occurs. And I think it's important to emphasize that it means that what the pancreatic beta cell is responding to is not just the presence of glucose in the gut lumen, but the active absorption of glucose into the bloodstream. And I think it's actually a really important way the enteroendocrine cells have adapted, is that the body is detecting glucose absorption, not just glucose that's present in the gut lumen. Next slide, please. And it seems that it's not only glucose that has adapted to be detected by absorption. So here I've shown you long chain fatty acids at the top and bile acids at the bottom, both of which also seem to need to be absorbed through the epithelium before they target G protein coupled receptors on the basolateral side of the L cells. So long chain fatty acids are detected by free fatty acid receptor 1, that's also known as GPR40. And on the bottom right, bile acids are detected by a GS coupled receptor, GPBAR1, that's also known as TGR5. So again, as I've said, the enteroendocrine cells don't just seem to be detecting what is passing by in the gut lumen. They seem to detect what is actively being absorbed. And this is why I say I've turned away from my original idea that this apical membrane surface is just acting as a chemosensor. I think it's important that the enteroendocrine cells are responding to absorption and not just as a passive detector. Next slide, please. So the big question is, could L cells potentially be targeted for the treatment of diabetes and obesity? And one of the major classes of druggable targets is G protein coupled receptors. So on the left-hand side is a picture of a differential gene expression in human ideal L cells of only showing the G protein coupled receptors. And you can see on the right of this are the L cells and on the left of the non-L cells. And for those who know the field well, I've highlighted receptors that we all know about. So at the top is free fatty acid receptor 1. Then there's GPR119, that's a long chain acyl-CoA receptor. GPBAR1, the bile acid receptor. GPR142 and CASR, those are amino acid sensors. FFAR2 is a short chain fatty acid receptor, and FFAR4 at the bottom is another long chain fatty acid receptor. So these are receptors that the cells normally use to detect what we've eaten, but could potentially be targeted with small molecular ligands to stimulate the L cells to enhance secretion of GLP-1. I've put, however, on the right-hand side, again, this image showing the distribution of hormones down the GI tract. And I've put it there to say that actually, even though the aim of this sort of therapy might be to increase GLP-1, it's not really going to be very possible, I think, to increase GLP-1 without increasing the other hormones at the same time. And that's because the cells that secrete GLP-1 are very overlapping with the cells that secrete GIP and CCK and secretin. And you could say that's a bad thing because you want to target only the L cells that secrete the GLP-1. On the other hand, I think you could say it's a good thing because many of these hormones have overlapping function. And if we're trying to mimic bariatric surgery, actually you may want to get to trigger secretion of an array of different hormones. And I think that's what we'll get if we start trying to target the L cells with any of these receptors on the left-hand side. So next slide. So one of the ambitions is to mimic bariatric surgery. So the bottom left shows the operation that's called a Roux-en-Y gastric bypass, and it's used to treat obesity, but it also, people who have type 2 diabetes and have a Roux-en-Y gastric bypass have a dramatic improvement in their diabetes. We've studied a group of patients who have a very similar operation, but they have it for gastric cancer, and they have prophylactic gastrectomy with a Roux-en-Y reconstruction. And the metabolic changes are parallel to those seen in obesity. However, since these patients are generally lean and young when they have their operations, the consequence of what is good for someone with type 2 diabetes is not so good for people who have it for gastric cancer, because anorexigenic actions and lowering blood glucose actually is quite detrimental in somebody who is otherwise lean and trying to maintain their body weight. So on the top right, you can see the plasma glucose changes after a 50-gram oral glucose tolerance test, and the plasma insulin and the GLP-1. And you can see that after the gastrectomy, insulin levels are dramatically raised, and you can see on the top right there the GLP-1s going up. Next. And to determine the contribution of GLP-1 to this insulin signal, we tested the effect of Exendin-9, which is a GLP-1 receptor blocker. And to the right of that, you can see that when we blocked the GLP-1 receptor in people who'd had a gastrectomy, it reduced the insulin secretion back down to normal levels. And next. And this is just to show you that actually it's not just a GLP-1 signal triggered, it's a GLP-1 and PYY. Next. And this arises, as I say, because PYY and GLP-1, and in this case, insulin-like 5, they come out of the same cells. And if you look at different parts of the GI tract, you could map different hormones in the same way. So on the left is shown an image of a primary colonic mouse L cell imaged by 3D super resolution microscopy. And 90% of the vesicles in the cell were triple labeled for 3 different hormones. And on the right, you can see the 3 different hormones coming out in very much in parallel in response to a range of different G protein coupled receptor stimuli. Next. However, there is some distinction between L cells. So this is a single cell cluster analysis from RNA sequencing data. And you can see 4 clusters of L cells. This is a combined colon and rectum from a mouse. Next. And And the way these L cells can be distinguished is actually that they are different depending on their location. So some are located more distally and some more proximally. Next. And in the other axis, there's a difference between immature and mature L cells. And I'm sure if we did this analysis further up the GI tract, there would be more different clusters appearing. Next. So I'd just like to finish with rather brief challenge for where we need to be moving in the future. So there's all these different gut hormones, and we know that gastric bypass surgery is a good thing, but I think where we need to be moving in the future is how can we mimic gastric bypass surgery by either targeting the gut or by mimicking the gut endocrine effects of bypass surgery without allowing our patients anywhere near the surgeons. And next. And finally, I would like to thank my group, particularly Frank Ryman, who co-runs it with me. In the colored boxes are people who are currently working in our group, and at the bottom right are people who have recently left our group but who contributed to the data that I've just shown you today. Thank you very much, and thanks for listening.
No, thank you, Fiona, very much for an elegant lecture. I wish we could hear some applause that it would deserve, but I guess one of the disadvantages, of course, of this format is you can't hear that, but it was really great. And it did also provoke a number of questions from people in the audience, and I have one or two myself, but let me just try to start with those that have come in from members of the audience. And the first one has to do with kinetics of response and the kinetics of glucagon versus GLP-1. Is glucagon faster? If we consider the gut in a postprandial state, would it make more sense for GLP-1 to be the faster hormone because it's located more proximal in the GI tract? So they really wanted to ask about how the location of the cells and the sensing has to do with the kinetics of response that we see in the blood.
So actually the first hormone to go up really quickly and dramatically is GIP, and that's located just distal to the gastric pylorus. And as soon as food starts to exit the gut, the GIP signal in the bloodstream goes straight up. For GLP-1, you need food to trickle a bit lower down the gut. So there aren't so many GLP-1 secreting cells in the top of the duodenum, but they start to appear a little bit lower down, particularly in the jejunum and the ileum. So you actually need a larger meal and a bit less quick digestion to get the GLP-1 signal. However, it does still start to go up within about 5 minutes of eating a meal. So it's quick, even though the cells are fairly low down. But we think that is because there are enough L cells, not right up in the very top of the duodenum, but high enough up that they can detect food almost as soon as it starts to exit the stomach. Glucagon, of course, is coming from the pancreas, so that's regulated in a slightly different way and requires absorption of nutrients before they can target the glucagon to change the glucagon signal.
Okay, great. A second question, which is one I had somewhat myself, was the comments you made about SGLT1 and its important role in the control of some of these secretions. And the question here was, since we use SGLT2 inhibitors, some of them also have some degree of SGLT1 inhibition, for example, canagliflozin versus dapagliflozin or empagliflozin. And they wanted to know that since GLP-1 release is mediated by glucose uptake, which can be inhibited by SGLT1, does that provide a rationale for preferring one of the SGLT2 inhibitors over another?
So actually, it's a very interesting finding, and one that hasn't been completely sorted out, because even though you block glucose absorption with an SGLT1 inhibition side of an SGLT1/2 inhibitor, you also, that means that you drive glucose further down the GI tract. And then the glucose comes into contact with more L cells further down. So depending on the extent to which SGLT1 is blocked, you can drive more GLP-1 further down where it might target the L cells in the lower gut. And then there's a slightly strange mechanism that isn't yet sorted out, that there may be some SGLT1 independent stimulation of L cells in the lower gut. It's been suggested that there may be bacterial fermentation to short chain fatty acids, which then target the L cells. But we don't really think that that's a strong enough signal to account for this, for the stimulation of L cells lower down the gut with SGLT1 inhibition. So I guess the answer is yes, depending on how much SGLT1 is inhibited, it can affect GLP-1 secretion, but it might not go the way you expect, because it might drive glucose.
It's delaying it to another site. And
yeah.
Different. Okay, and actually you already brought up in your answer, a couple of people asked questions about commenting. Would you comment on how does the system that you describe interact with the role of the gut microbiome and having effects on glucose homeostasis?
So it's a very interesting question. So there are undoubtedly products of microbial fermentation that affect enteroendocrine cells. So short chain fatty acids can enhance GLP-1 secretion. Indoles, which are products of amino acid fermentation, can affect enteroendocrine cells. And then lipopolysaccharides can affect L cells, and metabolites of bile acids can differentially affect L cells in the colon. So we think that there probably is some sort of background underlying influence of gut microbial products on the enteroendocrine cells in the colon. However, what has not really been resolved is if you kill off all the microbiota, it doesn't really have a dramatic effect on enteroendocrine cell function, at least acutely. So I think we're still in a bit of a difficult understanding. It doesn't look as if there's an enormous regulatory effect, but there may be some.
Okay, and maybe I'll take one last question before going forward, and that is a couple of people ask about also the crosstalk between stimulation of cytokines in the intestine and stimulation of GI hormones like GLP-1, and how do cytokines play any role in the changes following bariatric surgery?
There are probably some changes in gut inflammation following bariatric surgery. There's not a great amount of literature to suggest that cytokines themselves have a very strong influence on the gut endocrine cells, and we and other people have done sort of studies to see if the gut endocrine cells themselves are changed by the surgery. But if you do a transcriptomic analysis of enteroendocrine cells taken before and after surgery, the cells themselves are actually not really very affected. So we think that the major effect of the bariatric surgery is to change nutrient delivery to a different part of the intestine without actually particularly having a major effect on the enteroendocrine cells where they lie. So there may be small effects, but it's the delivery that we think is the major, the nutrient delivery is the major effect of surgery.
Okay, perfect. Well, there were several other questions. I'm sorry we don't have time to answer those, but maybe some of those can be addressed online. And I want to thank you for a really elegant presentation to start off the symposium, and now turn it over to my co-chair, Jeff Flyer.
Okay, thank you, Ron and Fiona. And it's really my pleasure to introduce the first of today's Alpert Prize winners, Joel Havner, who is professor of medicine at the Harvard Medical School, associate physician at the Massachusetts General Hospital, and a former investigator of the Howard Hughes Medical Institute. Joel is the director of the Laboratory of Molecular Endocrinology in the Department of Medicine at the Massachusetts General Hospital. Dr. Havner received his BS degree from the University of Redlands in Redlands, California, and his MD degree from the University of California School of Medicine in LA. His research interests are in the fields of obesity, diabetes, metabolism, with a focus on the interactions of growth factors and morphogens, on the expression of transcription factors during development, and in the regulation of hormone production by endocrine organs. He has authored over 450 research articles, books, and reviews on these subjects. In addition to his work leading to the discovery of the insulinotropic hormone glucagon-like peptide 1, his lab discovered cyclic AMP response element binding protein, the pancreas duodenal homeodomain protein, and the existence of multipotent stem cells in the pancreas. Dr. Habner is the recipient of many awards. These include the Edwin Astwood Award, the Robert Williams Distinguished Leadership Award, and the Outstanding Mentorship Award of the Endocrine Society, as well as the Harrington Prize for Innovation in Medicine, which he shared with today's two other prize recipients. This is from the American Society for Clinical Investigation. Today, we have the pleasure of listening to Joel tell us about glucagon-like peptide 1, discovery, structure, and activity. Joel, thank you.
Thank you, Jeff. It's a pleasure to participate in this elegant symposium. And I would just begin by liking to thank the Warren Alpert Foundation for this award. I'd also would like to thank the Harvard Awards Committee for their selection and their staff for organizing this symposium. Can you have the next slide, please? What I'd like to do in the time I have today is to describe the early discovery studies that we did in our lab and leading to the structure of the GLP-1 family of proteins and the early studies on their activity. And the next slide, please, shows a model of the pancreas attached to the intestine as just an orientation point to say that the two major hormones are made in the endocrine pancreas. That's insulin from the beta cells and glucagon from the alpha cells, and also the somatostatin in the delta cells. The insulin and glucagon are two hormones that work both together and in opposition. And insulin is known as the feeding hormone. It stimulates glucose uptake by peripheral tissues. And the glucagon is a fasting hormone that produces glucagon and increases glucose, increases glucose release during times when there's no nutrients entering the intestine. The point I'd like to make is back in 1978, when we began these studies, there was quite a bit known about the biosynthetic pathway of insulin, but virtually little information about how the hormone glucagon and actually somatostatin are made. So we decided that we would start and initiate studies to determine how and the pathway of these two hormones. So next slide, please. So as we determined to utilize a recently discovered technology that is called recombinant DNA cloning, which was developed by Drs. Herb Boyer, Stanley Cohen on the West Coast. And it obviates the requirement for having to isolate scarce proteins from large amounts of tissue, go through purification, and it expedites actually obtaining complete sequence of proteins rather than partial sequences that have to be knit together. And it involves isolating the mRNA, the messenger RNA, using a remarkable enzyme called reverse transcriptase to convert it into cDNA, and then decoding the DNA nucleotide triplet sequence code to determine the protein. And the next slide, please, shows the result of our initial studies. And you may ask, what is this menacing looking creature on the left side of the slide? Well, that's a Atlantic anglerfish, otherwise known as a monkfish. And there's a story actually goes with this. We had initially intended to isolate messenger RNA from islets and endocrine tissue that's was isolated from the rat and then human pancreas. But at the time we began the studies in 1978, we ran into some headwinds, namely a moratorium was placed on recombinant DNA research that closed down the research studies in Cambridge and was pending in Boston. But we got permission from the city council to go ahead with cold-blooded animal, not warm-blooded. So that hence we got the idea that we could get fish from the local ocean here that Boston is adjacent to, and succeeded in retaining the tissue that has the endocrine cells in it called the Brockman body. And that's shown on the right of the slide. And we went through learning the process of cDNA cloning and sequencing the nucleic acid. And at the bottom shows our initial discovery, and that is the sequence of the anglerfish cDNA for the messenger RNA. And actually there were two non-allelic sequences that were arrived at. And this is due to the hard work of Kay Lund and Richard Goodman in the lab. I found that it's quite interesting that the sequence of glucagon was actually contained in the anglerfish. We could determine that was glucagon due to the very close similarity to the mammalian glucagons that are already known. But as we went from the left to the right, we could see that there was another sequence that looked very similar to the glucagon, but it was not glucagon. And in both of the non-allelic sequences we had, there were two then GRPs, we call them, glucagon-related peptides. It's also a signal peptide at the N-terminus, which we're just beginning to learn is a requirement for the entry of the prohormone into the endoplasm, from the endoplasmic reticulum, where it's made in the cell, into the Golgi apparatus for packaging into secretory granules. We're named this as a pro-glucagon in parallel with the naming of insulin precursor to proinsulin. Next, so this shows that looking at the detail of the sequence of the GRPs, these new C-terminal peptides, and comparing them with different members of the glucagon superfamily of peptides, we seem to be a similarity to a peptide called GIP, which Fiona Gribble has just described as one of the intestinal peptide hormones. And it was actually subsequently named glucose-dependent insulinotropic polypeptide, and determined by Werner Creutzfeldt's group to be an incretin hormone, which we've already heard a little bit about from Jeff Flyer, that these are intestinal peptide hormones that augment glucose-dependent insulin secretion. So it gave us a suggestion in these very early stages of discovery that the peptide may be related to the glucoincretin GIP. Next slide, please. What became important next was to determine the sequence of the pro-glucagons of mammalian origin. And what you're looking at here is a diagram of a sequence determined by Heinrich and Lund and just Lana Mojal in my lab at the time, and also shows the gene structure. So the mRNA shows that has the glucagon, and remarkably two, but not one like the anglerfish, but two glucagon-related peptides, which were at the time then renamed glucagon-like peptides. So there was glucagon-like peptide 1, glucagon-like peptide 2, and so the three hormones encoded in the pro-glucagon of the rat, subsequently concomitantly actually shown in the guinea pig, and then the human by Graham Bell in Bill Rutter and Dan Steiner's labs. interesting, we'll see in a moment that the cleavage of the pro-glucagon into the hormones is markedly tissue specific, comparing the pancreas to the intestine. In the pancreas, the major hormone that's released, shown at the bottom here, is glucagon in the green on the left of the slide. Whereas in marked contrast in the intestine, the cleavages, enzymatic cleavages, result in the formation of the GLPs, the GLP-1 and GLP-2. This is a consequence of two different prohormone convertases expressed, PC2 in the pancreas and prohormone convertase 1,3 in the intestine. The next slide, please. Yeah, the remarkable, what you're looking at is a summation of many chromatographic column analyses, HPLC analyses of extracts from rat pancreas and compared to rat intestine, performed by Svetlana Mojov in the lab over, it took almost a year of many column runs and using newly produced antisera to the specific for the different peptides, and showed just what I showed you previously, that in the intestine shown on the left, the predominant peptides that we see are the GLPs in the red circle, and the unprocessed peptide called glycine contains glucagon, an inactive peptide. Whereas in the islets, pancreas, endocrine tissue, the endocrine cells, the glucagon is the major peptide that's released, and the GLPs are predominantly locked in a major pro-glucagon fragment, the biologic function of which is unknown at this point. However, due to the high sensitivity of the chromatographic technology, you could detect there were indeed GLPs produced in the endocrine pancreas at low concentrations. Next slide, please. So a summary of what I've just shown you of the glucagon-like peptide discovery and structure, that glucagon has two distinct glucagon-like peptides are synthesized in the form of a prohormone, which was named pre-pro-glucagon. And the multiple peptides are cleaved from pro-glucagon during biosynthesis by selective post-translational processing carried out by prohormone convertases. The GLPs predominate in the intestine and glucagon predominates in the pancreas. And then the next slide, please. Now we go to switch to the activity. One of the very first studies, early studies that was done that determined what of the peptides are active. And what we had found was that GLP-1 came in two varieties, a peptide of 37 amino acids and one of 31 amino acids that differed by a 6 amino acid deletion at the amino terminus of the peptide. So we call them GLP-1 1 to 37 and GLP-1 7 to 37. And they found that the peptides activity in insulinoma cells, these are in cultured cells, stimulates the formation of cyclic AMP. And it's only the 7 to 37 peptide that's the active peptide. The 1 to 37 was inactive. And that's shown in the upper panel. And then the lower panel showing that the active 7 to 37 peptide stimulates both the expression of the proinsulin mRNA, meaning stimulates insulin biosynthesis and also stimulates insulin secretion. The next studies that we carried out were, go to the next slide, please. Yeah, there we go. So it was in an intact rat pancreas, a perfused pancreas, a system that had been developed by Gordon Weir at the Joslin Diabetes Center. And with Svetlana Mojav's ran studies along with Gordon and his new perfused pancreas system, comparing 7-37 and the 1-37 GLP-1 isopeptides. And we found very nicely is that the 7-37 stimulated insulin secretion from the perfused pancreas, which is shown on the left, and the 1-37 peptide at 1,000 times higher concentrations had no effect. And the next slide shows another study of the activity of GLP-1, this time using a GLP-1 mimetic called Exendin-4, and in studies with Susan Bonner-Weir in a system that she had developed that causes type 2 diabetes in rats. That is the partial pancreatectomy. Removing 2/3 of the pancreas results in a state of diabetes that mimics human type 2 diabetes. And that the GLP-1 mimetic peptide at the top panel shows stimulated the lowering of glucose, plasma glucose, shown in the blue symbols, compared to the untreated control in the upper yellow symbols. But what was somewhat of a surprise, we looked at some of the other parameters of the pancreatectomized rats, was that the GLP-1 also stimulated the proliferation of beta cells, shown on the right, however, resulting in the increase in beta cell mass. So it actually not only increased insulin secretion and lowering plasma glucose, but increased the cells that produce insulin in the pancreas. Next slide, please. And some studies we did in collaboration with Josephine Egan at the National Institutes of Health, using now another model. It's a mouse model of diabetes called the DBDB, leptin receptor deficient mouse. And it has a type 2 diabetes model in animals. And here we again using the GLP-1 mimetic Exendin-4, EXD4, shown at the upper left, that the glucose is lowered by the hormone. It stimulates insulin secretion and also lowers the mouse human the equivalent of the hemoglobin A1C, meaning that the glucose lowering effects are preserved. And also a finding in these studies was that the GLP-1 mimetic stimulated the production of the pancreas transcription factor, PDX1, which is shown that is required for pancreas development. And this is shown on the Western blot, the protein level on the left, and also that it increases beta cell mass. So the mechanism of increased beta cell mass, and it seems to be due to the expression of the transcription factor PDX1. Next slide, please. Then in humans, a planet study in collaboration with my colleague David Nathan at the Mass General, showing that in type 2 diabetic subject volunteers, that the infusion of GLP-1 over 30 minutes, starting at the beginning of a meal, a test meal, has a rather remarkable effect on lowering blood glucose level at the top, which is shown in the solid symbols. That if with its control saline infusion, you can see the blood sugar goes up. It's typical of type 2 diabetes. Whereas with the concomitant infusion of GLP-1, blood sugar is actually lowered, but not lowered to below normal glycemia, which is an important finding that the administration of GLP-1 does not lead to hypoglycemia, as is seen with insulin. In addition, you can see at the bottom, the insulin levels are stimulated by the GLP-1. Next slide, please. In summary of the studies of the activity, that the GLP-1 stimulates glucose-dependent insulin secretion, lowers plasma glucose in diabetes, and in the animals studies without resulting in hypoglycemia. So that there's a glucostat effect in the beta cells, that when the blood sugar level gets down to approximately 60 deciliters per milligrams per deciliter, the effect of stimulating insulin lowering glucose is lost. And it's also, the studies reveal that the GLP-1 acts on a cyclic AMP coupled receptor and induces insulin biosynthesis, stimulates beta cell proliferation and survival, and increases beta cell mass, increases the PDX expression. Next slide, please. We go on to the, let's see, yeah, I want to just briefly show a series of studies that were done by my postdoc at the time, Sengu Liu, where we compared the effect of a number of beta cell damaging agents on the repair effect due to the stimulation of the expression of GLP-1 in the alpha cells of the pancreatic islets. So what it does, I'm going to show you how the administration of either a chemokine, SDF1, or a mixture of cytokines which damage beta cells, switch on the production of the PC1-3 convertase, leading to, in alpha cells that make glucagon, leading to the expression of GLP-1. The next slide, please. This is shown here on the left with if we give the either on the left the PC, we're looking at the PC1-3 expression in mouse pancreatic islets, and the increase in PC1-3 in response to the chemokine SDF1, which is expressed by the beta cells in response to damage by either cytokines or SDF1, showing on the right, which is the GLP-1 concentration in the islets goes up over a period of 1 to 4 hours. Next slide, please. This is just a model of what I've just shown you, the data, and that the injury of beta cells over here on the left increases the expression of chemokine SDF1, which then acts on the adjacent alpha cells in the endocrine pancreas to increase PC1-3, which turns on the expression of GLP-1, which then acts on the beta cells to increase growth and survival. Next slide, please. Just a model showing the summary of the diverse actions of GLP-1 on many different organs of the body, and it's leading to the future directions of the work that we're currently involved in, which is shown in the next slide. That is that the selective enzymatic cleavage of GLP-1 from proglucagon is not the end of enzymatic cleavages, but that GLP-1 itself is actually cleaved by selective endopeptidases to create GLP-1 derived peptides, which are novel, that seem to be novel, distinct bioactive peptides that are receptor independent actions, and the evidence would suggest that they're not degradation products. And this is shown here. It's the cleavages, which you'll probably hear more about from Jens Holst, removing the N-terminal 2 amino acids with an endopeptidase DPP4, but then subsequent cleavages at the C-terminus, which generate small peptides like a nonapeptide and pentapeptide. And the next slide, please. Which in some studies that have been going on over the last several years, a number of bioactivities have been attributed to the nonapeptide, such as the modulation of oxidative phosphorylation, fatty acid oxidation, gluconeogenesis, energy expenditure, oxidative stress, and apoptosis. And these are all functions mediated by mitochondria. And it's shown that the peptide is exported, imported into cells, presumably by a cell penetrating process, and then targets the mitochondria where it regulates these processes. So it's shown in a very nice recent study from Mansour Hussein's lab in Toronto, demonstrating that the nonapeptide in mouse models of heart attacks has salubrious effects on post-ischemia reperfusion injury. Next slide, please. I'd like to now acknowledge and thank all of my collaborators and co-workers over the years, and I give a special thanks to my long-term mentor and colleague, John Potts. Thank you for your attention, and that's it. That's it.
Okay, well, thank you, Joel, for a wonderful talk. And I will follow up with one question that several people have asked. Running a little behind, so I think I may limit it to the one question, and that is, I know from very early on in your studies and others, there's been a lot of interest and discussion in what could be the beneficial effect of the stimulation of beta cell hypertrophy by GLP-1 and various therapeutics based on it. And do you feel that we know currently whether this occurs in vivo in humans? And does it suggest anything about the approach to using drugs that agonize the pathway?
Well, I think we don't really know as far as I can tell, but I think that's a very good question that deserves pursuit. And I think there's also some questions that since the experiments in animals would suggest that the GLP-1 is actually a growth promoting factor with respect to beta cells. And beta cells come from duct cells. They differentiate. They can also come from transdifferentiation of alpha cells. So one of the speculations at this point is that the GLP-1 is actually activating developmental pathways that are actually in place during the formation of beta cells during embryogenesis, development of the embryo. So there's also possible potential for neoplastic transformations, which has not happened at this point. So it seems to be a safe treatment, safe medication without any cellular side effects.
Okay, thank you, Joel. And I would now like to turn it back to Professor Khan to introduce the next speaker.
Thank you, Jeff, and thanks, Joel. And it's my pleasure to introduce the third speaker of the symposium and one of the 3 recipients of this year's Warren Alpert Prize, and that's Jens Juul Holst. Jens Holst received his MD degree from the University of Copenhagen in Denmark, and he is still currently at that institution as a professor in the Department of Biomedical Sciences and a group leader for translational metabolic physiology at the Novo Nordisk Foundation Center for Basic Metabolic Research at the University of Copenhagen. Jens heads a research group focused on hormones that regulate metabolism and appetite regulation in obesity and diabetes, and especially how these play a role in the pathophysiology of disease, as well as potential treatment of disease. As pointed out by Jeff in his introduction, and actually as I frequently point out to my own fellows who are doing research, that usually when something is going on in one lab, there's something going on somewhere else that approaches the problem from a different approach, but comes to a very similar conclusion. And in this case, while Joel and his colleagues were working to identify through cloning and molecular biological approaches this novel family of hormones, Jens Holst and his colleagues were really looking in the blood to try to identify what was the form of circulating glucagon, or what were the forms of circulating glucagon in human blood that might be biologically important. And in this process, they discovered a form which they initially termed enteroglucagon, which we now know as GLP-1, which was related to glucagon, but clearly different, and was coming not from the pancreas, but from the gut. His group showed that a truncated form of GLP-1, as Joel also pointed out, is actually the most potent, a very strong potent insulin secretagogue in vivo, and that it's a glucose-dependent secretion, that is, it's acting as an incretin. Jens and his colleagues then went on to describe the critically important roles of GLP-1 to lower blood glucose in type 2 diabetic subjects and to reduce appetite as well. As a result of all of his work, Dr. Holst has been the recipient of several prizes, including the 2017 UC PH Innovation Prize, the Harrington Prize for Innovation in Medicine from the ASCI, the Nordic Fenstrom Prize, the Anders Jahr Senior Medical Prize, the Marie and August Crow Award, the EASD Award, the Bager Sorensen Award, and also the KFJ Prize of the University of Copenhagen. Jens has also helped translate some of his findings through formation of a couple of startup companies, which he was a co-founder. Today, he's going to tell us about his remarkable discoveries of the role of GLP-1s in humans. Jens?
Thank you very much for this, Ron. And thank you very much to the Warren Alpert Foundation for making all of this possible. I've really been looking forward to this. And now I will talk a little bit about GLP-1 in humans. Next slide, please. So in Copenhagen, we were interested in postprandial reactive hypoglycemia and trying to find out how that could happen. And we had an idea. We were working with the incretin effect in the late 70s. And so we thought that it might have something to do with the incretin effect. Next slide, please. And we were interested in these cells that you can find in the gut epithelium in humans here. And what they are endocrine cells that react with antisera towards glucagon. So could they be responsible somehow? And this was an idea that came from the work of Symposium and Marx, because they had shown that glucagon was able to stimulate insulin secretion. Next slide, please. So the tool we were using, I've had a lot of fun with this tool. I really like it. It's the perfused pancreas, and this is a pancreas from a pig. And it's doing all that a pancreas is supposed to do. Right here, I've been stimulating the vagus nerves. So they are mounted in that electrode you can see. And the picture is taken 1 minute after electrical stimulation of the vagus nerve. And you can see that it has produced a lot of pancreatic juice. I really love that model. But what we did was then to put gut extracts that were enriched for glucagon-like peptides on this preparation. And next slide, please. Eventually, we were able to find one which really, really, really stimulated insulin secretion very, very powerfully, as you can see here. Next slide, please. So eventually, by painstakingly going through peptide extraction and sequencing, we were able to work out what the different peptides were. So all of these peptides were actually extracted and sequenced, and we came up with a structure for GLP-1 of 78 to 107 amide. It's actually an amide. And we also pulled out GLP-2 actually in 1988 and showed what the correct structure of that was. And all of these references you can see down there below. So, and you've already seen the differential processing in the talk by Joel Havener. Next slide, please. So we had found an insulin-stimulating peptide in the gut. And that was not really dramatic in those days because there were so many. There were peptides stimulating insulin secretion all over the place. And it turned out that most of the peptides, they did something to insulin secretion if you gave enough of them. What really turned the picture was this. Here we're looking at the new GLP-1 on glucagon secretion from our perfused pancreas. And here you see something which was unusual. It strongly inhibited the secretion of glucagon. And this is important. Next slide. Because if you infuse this new peptide in absolutely physiological concentrations, as you can see in the lower panel, what will happen is that you will inhibit glucagon secretion and you will stimulate insulin secretion as shown in the upper panels. And as a result, you will inhibit hepatic glucose production by about 30%, as shown in the third panel. And there you can see what happens. So next slide. As a result of those changes, the glucose level in plasma will start to fall during these absolutely physiological experiments here. But they don't fall very much. They only fall by about 1 millimoles at the most, regardless of how long you continue the infusion. But this is what actually happens. Next slide. So eventually, precisely this process turned into this fantastic observation here that we were able to do in a collaboration with Michael Nauk in 1993, where people with type 2 diabetes who were admitted to hospital because they needed insulin therapy were infused intravenously with GLP-1, as you can see, for 4 hours. And during the course of that, in the top panel, you can see that their blood glucose levels, which were really high, came down to absolutely normal levels. And that middle panel occurred while their insulin secretion was stimulated, and bottom panel, their glucagon secretion was inhibited. And as you can also see that as the glucose levels are approaching normal levels, the stimulation of insulin goes down again, and the inhibition of glucagon goes up again. So this was the glucose dependency of these processes. This really was the turning point. This was the decisive effect of GLP-1 in type 2 diabetes, which really started the whole thing off. Next slide. We had been working with the gastrointestinal actions of glucagon for quite a while, and we knew that glucagon might inhibit gastric emptying and also inhibit appetite and food intake. So it was natural for us to start looking for this with a new glucagon-like peptide from the gut. Next slide. So here in the early 90s, 93, here is this first study. We infused people with a truncated GLP-1, and what we found was that it inhibited gastric secretion and pancreatic secretion and gastric emptying. And that's very clearly shown in this graph. Next slide. And we went on to look at food intake and appetite. And here you can see what happened when we infused again in physiological amounts GLP-1 into people and looking at the energy intake at limited meals. And you can see that there was a significant inhibition of food intake. And also in the intermeal period to the right, you can see that satiety is enhanced and hunger is suppressed. So this was this new interesting effect of GLP-1. Next slide. I can't resist the temptation to show you the current status of that effect. So this is a study where the new GLP-1 agonist, GLP-1 receptor agonist semaglutide was given to people weekly injections over a period of 68 weeks. And you can see that there was a weight loss of no less than 18%. So this has moved from an interesting observation that it was a physiological regulator of food and food intake and obesity into real anti-obesity therapy. This is a remarkable evolution, I think. Next slide. So regarding GLP-1 in normal physiology in humans, there's no doubt that this is an upper intestinal GLP-1. The upper intestinal GLP-1 functions as an incretin hormone. I'll come back to that. In the distal small intestine, I think the main actions of GLP-1 is to act as an enterogastrone that signals nutritional abundancy, and that it produces satiation and limits further food intake and promotes nutrient disposal if anything comes in, and it does that via stimulation of insulin secretion. As Fiona mentioned, the colonic GLP-1 is much more difficult to find out. We think perhaps right now that it serves a local mucosal protection, and I must say that if you take away the colon, as you can do in people, total colectomy, the meal GLP-1 responses are completely preserved. They're completely identical. So you can't see the colonic secretion of GLP-1 at all. Next slide. So when we put all this together and look at the type 2 diabetic phenotype to the left and all the actions of GLP-1 to the right, you get this fantastic picture where everything that is wrong in type 2 diabetes can be somehow remedied by the actions of GLP-1, whether it's the impaired beta cell function, and you've already heard about that, the reduced beta cell mass. I'm not so sure that that is actually happening in people, but perhaps the beta cells are protected in the long run, and I think this is probably possible. We have the glucagon hypersecretion, and we have the inhibition by GLP-1. We have the overeating and obesity, and we have the strong effects of GLP-1 on food intake and the weight loss. The cardiovascular effects, I'm sure that Dan Drucker will talk a lot about that. And even insulin resistance is also improved, but that is probably secondary to the weight losses. So all of this came out in the proof of concept study that we did in the Lancet in 2002. And that's why we called it the diabetologist's dream. But was there a snake in paradise? Next slide. Yes, there was a snake in the paradise, because the native GLP-1 molecule is rapidly degraded by dipeptidyl peptidase 4, the enzyme, and also cleared in the kidneys extremely rapidly. And here you can see the cleavage site, and the fragment, the metabolite, is inactive. And that happens with a half-life of 1 to 2 minutes, incredibly rapidly. Next slide. So if you inject people with type 2 diabetes with a maximum tolerated dose of GLP-1, here 1.5 nanomoles per kilogram, you'll see that the black line shows you how much survives in the intact form and how much is then present in the form of the inactive metabolite. So it's a waste of good peptide to inject it subcutaneously to people. Next slide. But looking at that, we felt that this was an interesting observation and that something could be done about it. So while looking at this degradation, we said inhibition of dipeptidyl peptidase 4 may prove a useful adjunct in the management of type 2 diabetes. And we were inspired, of course, by the use of ACE inhibitors to treat hypertension. Nothing special about that, but this is what we proposed. And we not only proposed this, next slide, but we went on to do experiments in pigs with an available DPP-4 inhibitor, Vagal and pyrrolidine, and we could give it after a GLP-1 infusion to see what happened with the second GLP-1 infusion, and that's what you see here. The red curve shows total GLP-1, or mainly the metabolite. The black curve shows the intact surviving active GLP-1, and you can see that the extensive degradation by DPP-4, even when you infuse GLP-1 constantly. And then we give the inhibitor, and now you can see during the second infusion, everything is protected. It really, really works. Next slide. And as a result of this protection, the stimulation of insulin secretion during a small glucose infusion here was tripled after the DPP-4 inhibitor. So this really proved that you can inhibit the degradation of GLP-1 with a DPP-4 inhibitor, and that you can greatly improve the actions of GLP-1 with a DPP-4 inhibitor. We're quite proud of this observation because it had such enormous consequences. Next slide. So summarizing about the DPP-4 inhibitors today, that's one thing, they're orally available. They're very effective as anti-diabetic medication in combination with metformin. You can provide 24-hour inhibition with all of the available compounds. They're used worldwide. And in many countries, they are now outperforming the sulfonylureas. And the best of the whole thing is that they have a very benign side effect profile. And there's even one more new piece of good news, that is that they will soon come off patent and they will be cheap. That's a sensation. Next slide. So how is it that this wonderful hormone can do anything at all if it's broken down immediately by DPP-4? And here you see an L cell in the bottom, in the villus of the small intestine, and it secretes its GLP-1. It diffuses across the lamina propria, finds the capillary, and gets degraded, so that only a quarter of what was secreted reaches the liver, where another set of DPP-4 cleaves half of what is offered, so that we are now back to about 12 or 10%, reaching possibly the pancreas. But what can happen is that the GLP-1, while it's still intact, can interact with afferent sensory nerves of the vagus system. And here you can see them in the black lines, that the sensory neurons, and they have their cell bodies in the nodose ganglion and signal to the brainstem and also to the hypothalamus, so that new efferent activities can be set in motion. So this is how it can actually work. Next slide. So if you ask where are the receptors, then here is a picture of the receptors for GLP-1 in the myenteric ganglion. And see how beautifully these nerve fibers wrap all the other cells here. It's really an impressive pattern of GLP-1 receptor expression. Next slide. So does this happen in people? Can you investigate that? And what we did was, I have this past as a surgeon, so I've done one of these vagotomies when I was a very young surgeon. So this is a truncated vagotomy, and there are a few patients out there that exist still with truncated vagotomies. So next slide. One of the ways to look at the effects of vagotomy is to look at pancreatic polypeptide responses to meal ingestion, because pancreatic polypeptide is extremely sensitive to actions of the vagus. And in the two lower panels, you can see a control experiment, and the black boxes are with infusion of GLP-1, and you can see that GLP-1 almost completely inhibits the PP response. And then if you look at the vagotomized individuals, that's the two upper curves, you can see that now after vagotomy, the GLP-1 has completely lost all of its effect. Next slide. And here we're looking at gastric emptying, same patients, and the two lower curves show the controls, and you can see the powerful effect of GLP-1 on gastric emptying. The two upper panels show what happened in the vagotomized individuals. Nothing. It lost all of its effect. Next slide. And here comes the important thing, the food intake. So to the right, you see what happens in the controls. You have the inhibition, 15% or so of the food intake to an ad libitum meal, whereas in the vagotomized individuals, absolutely nothing happened. So again, GLP-1 lost all of its effect after the vagotomy in these individuals. Next slide. So this was interesting, of course, and we, like Fiona talked about, we're also interested in whether GLP-1 could explain some of the effects of Roux-en-Y gastric bypass. So first of all, we looked at some patients and gave them the GLP-1 receptor antagonist, exendin 9. And sure enough, as you can see in the light gray column, there's an increase in food intake if you block the actions of GLP-1. But then we looked at them after they had had their Roux-en-Y gastric bypass, and then there was no effect of exendin 9. And this was quite a disappointment and a surprise to us. But it turned out, if you look to the right panels here, that what happens is that not only does the operation tremendously increase the secretion of GLP-1, the green lines, the full green lines, but also the secretion of PYY, as we heard about. But the use of exendin 9 even more increased these levels of GLP-1. So now you had a tremendous increase of not only one of these hormones, but also the other one. And both of them are powerful suppressants of appetite. Next slide. So what could we do? Well, GLP-1, we could also block it, always block it with exendin 9, of course. But for PYY, there was no antagonist. But PYY is secreted in a form of 1 to 36, and it must be activated by DPP-4 to create PYY 3 to 36. That's the active form. And that could, of course, be prevented with a DPP-4 inhibitor. So in this way, we could block the actions of both GLP-1 and PYY. Next slide. So what we did was to look at meals. And here you can see with the exendin, we could block the PYY. We could block the GLP-1, and with the DPP-4 inhibitors, we could block the PYY effects. And we could use both of them to block the entire effect. And next slide. So yes, indeed, giving only one of them, exendin 9 or DPP-4 inhibitor, did not really significantly change food intake. but giving the two of them caused an increase in ad libitum food intake by 20%. So proven case, it was indeed the secretion of GLP-1 and PYY together that is the cause of the decreased appetite and food intake after the Roux-en-Y gastric bypass. Next slide. So finally, what about the incretin effect? What happened to that? Well, a very recent development has enabled us to look at this in more detail. So we have the exendin 9 antagonist to block the actions of GLP-1. But now in our laboratory, in the laboratory of Mette Rosenkilde, we were able to develop an antagonist against the actions of GIP that could be used in humans. That is GIP 3 to 30 amide. And here we're looking at glucose tolerance after an oral glucose tolerance test with the use of these antagonists. So the bottom curve shows the control experiment, and then you can see that each of the two blockers impair glucose tolerance as expected, but the combination of the two even more impairs glucose tolerance. That's the top black curve. And to the right, you can see the AUCs of these curves so that you can see the impairment even more clearly. Next slide. And that, of course, had to do with insulin secretion. Now, up here, you see the insulin secretion and the C-peptide levels. But since glucose levels were also changed by this antagonist administration, we had to do something. So at the bottom panel, we've calculated C-peptide to glucose ratios to correct for the changes in glucose. And in the red circle, you can see that ratio, that is a kind of insulinogenic index, and you can see how strongly it is impaired by the two antagonists, particularly in combination. So next slide. Our friends, Michael Nauk and Joris Meyer, then sat down with a calculator and did some calculations from the various columns and came up with this result, that in humans, if you, from experiments where you could block the individual contributions of the incretin hormones, GIP was responsible for 44% of the insulin secretion. And Michael Nauk was very happy about that because he had predicted that all the way through since he started in 1899. And glucose was responsible for 33% of the increase, whereas GLP-1 was responsible for 22%. So now we know what it looks like with these incretin hormones in people. Next slide, please. So that's my concluding slides. What can we say about GLP-1 in humans? Well, it is a hormone that is secreted in relation to meal intake, and there's no doubt that this is the most important thing. It's not a fasting hormone. It's an incretin hormone which is responsible for at least 20% of the postprandial insulin response, and in addition, it inhibits glucagon secretion, which is probably very important. It is also an important enterogastrone hormone regulating appetite and food intake, upper gastrointestinal secretion, gastrointestinal motility, and then it acts importantly via afferent and efferent vagal mechanisms. So that's what I wanted to say today. But first of all, next slide, I have to mention and thank all the numerous contributors to all of these projects, and I've listed them here so that you can see all the names. Thank you very much.
Okay, so thank you, Jens, for a great talk and a lot of even very new data. We have a few questions from the audience. We'll take a couple of them. One of them really relates to the use of DPP-4 inhibitors and the question about DPP-4 being a relatively non-specific protease, and therefore it could, in its inhibition, affect many peptides which control food intake. And how can you interpret DPP-4 vis-a-vis GLP-1 or other hormones specifically?
If you recite to rodent studies with knockouts and so forth, there are actually some beautiful studies indicating that the main effect on metabolism is via GIP and GLP-1. So that's one way of doing it. We have been very active in trying to look at antagonists like exendin 9, and now we are also using the GIP 330 to identify precisely how much of the actions are due to DPP-4 inhibition. But the fact is that GLP-1 is an extremely sensitive substrate for DPP-4. And that really explains that it is so extensive, that GLP-1 is so extensively degraded. And for each of the potential substrates, you really have to work out the physiology of it. So you need to go to people and see how much of this potential substrate is actually degraded by DPP-4 in vivo, and how much, what does that mean? So we've done that for GLP-1 and DPP-4, but it needs to be done for every substrate. There's a good case for GIP also. We've also worked through that. So it really does protect GIP. GIP does not do a lot in type 2 diabetes, we think. It may, the effect may be improved upon prolonged DPP-4 inhibition, but not a lot.
Okay, and that really gets to a question I was also thinking about, which is GIP is a very potent incretin.
Yes.
And yet it doesn't do much in diabetes compared to GLP-1. Could
That's
you
right.
comment on the balance and how these, how this plays out, that it doesn't have more effect?
So it's a very interesting question, particularly these days where there's a big debate going on as to whether one should combine GLP-1 and GIP, or whether one should combine a GLP-1 with a GIP antagonist. And the reason for that is that a lot of data suggests that GIP promotes obesity rather than the opposite. So there are a lot of ideas out there, and there are, as I'm sure you know, there's a GIP-GLP-1 co-agonist right now in development, which has very profound effects on obesity and on diabetes also. It's called tirzepatide, developed by Eli Lilly. But there are a few publications, very, very recent ones out there. We have contributed to some of them, where you can see the discussion on what really goes on here. And my idea is that the tirzepatide is at least a very potent GLP-1 agonist. And this is one of the secrets behind its activity. I am much more doubtful about the GIP contribution.
All right, very good. Well, thank you again for an excellent presentation and of your lab's work and contribution in this area. And I'm going to turn the program back over to Jeff Flyer.
Okay, thank you, Ron, and thank you, Jens. It is now my pleasure to introduce today's third Alpert Prize winner, Dr. Daniel J. Drucker, who I first had the pleasure of meeting in 1979 when he was a medical student in Toronto who took an endocrine elective at the Beth Israel Hospital shortly after I joined the HMS faculty, and I could predict his future success from that moment. Dan is an endocrinologist and professor of medicine in the Division of Endocrinology, Department of Medicine at the University of Toronto, from which he received his MD degree. He received training in internal medicine and endocrinology from the Johns Hopkins Hospital in Baltimore and the Toronto General Hospital at the University of Toronto, after which he came to Boston to do a research fellowship in molecular endocrinology at the Massachusetts General Hospital. And then after completing that, in 1987, he established his own research laboratory in Toronto. Dan is a pioneer in diabetes treatment as well as pathophysiology. His work has provided important insights leading to the development of new drugs for the treatment of type 2 diabetes. Currently, his laboratory at the Lunenfeld Tannenbaum focuses on understanding the molecular biology and physiology of glucagon-like peptides. Dan is the recipient of the Clinical Investigator Award from the Endocrine Society, the Banting Medal for Scientific Achievement from the American Diabetes Association, the Manpei Suzuki International Prize for Diabetes Research, and in 2015, he was elected Fellow of the Royal Society of London and was also appointed as an officer of the Order of Canada, which sounds really important. Today, Dan will be telling us about the protein peptide GLP-1 from metabolism to inflammation and cardioprotection. Dan.
Thanks very much, Jeff. So I'd also like to thank the Warren Alpert Foundation for their vision in regard to the importance of recognizing and supporting biomedical science. And they do much more than that, and they're really a terrific organization. And as well, thank you to you and Ron and your colleagues who put in large amounts of work year after year and looking through the Alpert Foundation nominations. And really, I'd like to thank the people in my lab who have done a tremendous amount of work and allowed me to learn from them and bring some of the science to you today. So next slide. So I've worked with a number of companies over the years, and I think it would be very clear to all of us that we wouldn't be here today if it wasn't for this tremendous collaboration from the bench to the bedside and the development of new molecules by numerous individuals working at companies. And these are my current disclosures, but people who have worked in this field to bring these molecules to market, you should also be very pleased with your contributions to the field. Next slide. So, I'd like to thank Joel Habner for giving a very young, inexperienced, endocrine trainee the opportunity to work in his lab. And the studies that I did, you can see on the left, we didn't really know what GLP-1 did. And simplistically, we thought cyclic AMP might be important given the glucagon story. And it turned out that if one used islet cells, we saw glucose-dependent insulin secretion and an increase actually in insulin gene expression. And Joel really had the vision to move the story forward, and here we are today. Now, a lot of credit goes to John Eng, who isolated this amazing lizard venom peptide, exendin-4, and no one was interested in talking to him. He patented this. No one wanted to talk to him or license it. And ultimately, he also was a pioneer in bringing to what we now know is the first approved GLP-1 receptor agonist to market. And we wanted to know if humans had exendin-4. Eugene Chen cloned exendin-4 from lizard. That's another interesting story about how we got the live lizard. And lizard exendin-4 was very different from human proglucagon, and we never could clone human exendin-4. This was before the Human Genome Project, obviously, and we now know why. So next slide. So when we continued the story to looking at GLP-1 secretion and degradation, and Jens beautifully introduced the concept of DPP-4, and we were brought into this field by Bill Bachochin, a chemist at Tufts University, who had companies developing DPP-4 inhibitors for the treatment of cancer. We were also contacted by some immunologists in France who made a DPP-4 knockout, and they could not find any major immune phenotype. But we were fortunate enough to take a look at the metabolism in these mice, and you can see that these mice had improved glucose, more insulin, more GLP-1, more GIP, and the important thing is they were healthy. Many people were very worried about blocking or inhibiting DP4. And here you can see this mouse that had no DP4, and it really was quite healthy and lived a very healthy long age. Now, Jens beautifully introduced the concept that one could use, mouse genetics to understand how DP4 inhibitors lower blood glucose. And here you can see the studies that were done by Tanya Hansadia and Lori Badger in the lab. And if you take away both the GLP-1 and GIP receptors genetically in mice, you completely lose all glucose lowering. So even though there are many substrates, it's really all GLP-1 and GIP, and I think this is held up in humans. Next slide. So a big surprise to us came when Louise Scrokey made the GLP-1 receptor knockout mouse, and we have to thank Bernard Thorens for cloning the GLP-1 receptor. So perhaps the defect in insulin secretion and the elevated postprandial glucose is not surprising, but look at the fasting glucose. It's elevated, and we know if we infuse exendin 9 to 39 into small animals or monkeys or humans, we see this glucagon effect, and glucose goes up because we're affecting glucagon secretion and no longer inhibiting it. So this was really a surprise, and this mouse has now been very valuable to us and many investigators in trying to understand the physiology of endogenous GLP-1 action. Next slide. So we know that in mice, GLP-1 and GIP are important, and that's the classic incretin biology that Jens reviewed for us, and certainly DP4 inhibitors act to potentiate those substrates. But DP4 is expressed in many different cell types, and actually soluble DP4 is pro-inflammatory. And there's a lot of literature suggesting that soluble DP4 might activate inflammation in many different tissues. Next slide. A surprise to us came when we treated animals with DP4 inhibitors. And you can see in the left-hand panel, and we've done this with many different DP4 inhibitors, but when you inhibit DPP4, soluble DP4 rises. And we also see some degree of increased inflammation in some of the tissues in these mice. So we looked with the collaboration of Rory Holman and the TICOS investigators, what happens to soluble DP4 in humans before and after treatment with sitagliptin. Now, these are people with diabetes, obesity, and cardiovascular disease. So they have a high tone of meta-inflammation. And really, there was no consistent direction for soluble DP4. In some people, it went up. In some people, it didn't change. And in some people, it actually went down. Next slide. And there was a lot of hope that the DP4 inhibitors would reduce cardiovascular events in the outcome trials. And that was largely based on animal studies and some human data that suggested that DP4 inhibition reduced inflammation. However, we had the opportunity to look recently, again at the TICOS trial, in over 600 patients. And shown here are some of the biomarkers of inflammation in people treated with and without sitagliptin. And the reality is there really is no meaningful directional change of plasma biomarkers of inflammation in humans with diabetes, obesity, and cardiovascular disease who are treated with sitagliptin in this instance. Next slide. So one of the controversies in the field that's become very interesting recently is how much GLP-1 is there in the pancreas relative to the gut, and how important might this be in talking to GLP-1 receptors locally in the pancreas. And the answer to this question really depends in part on what models you use to interrogate the question and what reagents you use to try and ask these questions. Next slide. So we and others have looked at the amount of GLP-1 in the pancreas. You may notice that Professor Gribble actually did show you some LCMS data and showed very, very little GLP-1 in the pancreas. And we have used some assays as well to look at mouse and human pancreas. And we see detectable GLP-1 in the pancreas, but it's at extremely low levels compared to the amount of glucagon. However, we know that the affinity of GLP-1 for the GLP-1 receptor is much higher than the affinity of glucagon for the GLP-1 receptor. So it still remains possible that under some circumstances, pancreatic GLP-1 might contribute. Next slide. So one way to answer the question about whether pancreatic GLP-1 or gut GLP-1 is preferentially important for physiology is to simply get rid of gut proglucagon and GLP-1 using mouse genetics. So we've eliminated the proglucagon gene from the entire gut selectively or just from the distal gut. And when you eliminate GLP-1 from the entire gut, circulating GLP-1 basically disappears. However, we see some modest metabolic phenotypes, but no change in food intake and body weight. In fact, we also see some modest metabolic phenotypes when we eliminate distal gut GLP-1, but again, no change in food intake or body weight. And this suggests to us that it's really brain GLP-1 that's likely controlling physiologically food intake and body weight, but obviously pharmacologically, we can get tremendous reduction of food intake and weight loss. Next slide. So I've tried to show you here some of the concepts that have derived from understanding the GLP-1 physiology, both in mice and humans. And I don't want to pretend that we have all the answers in the pancreas in the brain. I've said clear mechanisms, but I think there's still some work to be done. But many of the actions that we know are transduced by GLP-1 in the other tissues still are not very clearly understood. And let's take a look at that. Next slide. So what about the brain and food intake? If we eliminate the GLP-1 receptors from the hypothalamus and the brainstem and the enteric nervous system with this Wnt1-Cre driver, we completely lose the ability of GLP-1 to inhibit food intake and to reduce body weight. So clearly, GLP-1 receptors in neuronal populations in those areas are absolutely fundamental. And we've done this with exendin-4, liraglutide, dulaglutide, same results. Next slide. But very interestingly, Jens highlighted the importance of the vagus nerve in transducing some of these signals. And so one can eliminate autonomic nervous system and nodose ganglia GLP-1 receptors using the FOX2B Cre driver. And on the left, you see the control results, very nice weight loss with dulaglutide. But notice what happens if we take out these autonomic and vagal GLP-1 receptors. Dulaglutide still works because we still have the hypothalamic and brainstem GLP-1 receptors, but it doesn't work as well. And this really suggests to us that there are multiple different regions of the nervous system that can transduce these GLP-1 receptor signals, ultimately converging on reduced food intake and weight loss. Next slide. So what about postprandial lipids? We haven't seen that discussed very much today. Both DP4 inhibitors as well as GLP-1 receptor agonists profoundly lower chylomicron secretion from the gut enterocytes, yet there are no GLP-1 receptors on gut enterocytes. So how does this communication happen? Well, one possibility is the brain. So if you take a look again, we can use the Wnt1-Cre system to eliminate the central nervous system and enteric nervous system GLP-1Rs, and we can use FOX2B to eliminate the autonomic nervous system GLP-1Rs. Yet when we do that, we still see that multiple GLP-1 receptor agonists still lower triglyceride excursion after olive oil loading. So we've eliminated almost all of the neural transmission mediated by GLP-1 receptors. There are no GLP-1 receptors on enterocytes, yet we're still seeing this profound suppression. And so this is still a mystery to us, and we're still working on it. Next slide. So what about the heart? In 2008, we had the FDA guidance and the rosiglitazone concerns. And at that time, we started working right away with Mansour Hossain. What did GLP-1 do to the mouse cardiovascular system? And in 2009, we said, it's cardioprotective. We see fantastic reduction of infarct size and improved survival. And then subsequent studies done by John Usher in the lab said, well, how does this happen? Is this due to the cardiomyocyte GLP-1 receptors? And John eliminated these with myosin heavy chain Cre, yet still saw profound cardioprotection, reduced infarct size, and improved survival. So it's clearly likely a combination of some other cell types in the heart that express the GLP-1 receptor, as well as perhaps some indirect effects. And I hope to have more to say on that in the coming months. Next slide. So interestingly enough, we had to wait several years until we saw whether this mouse biology was in fact reflective of what happened in humans. And we now know from multiple cardiovascular outcome studies that long-acting GLP-1 receptor agonists reduce cardiovascular death. And that's been, I think, extremely important for the field of diabetes and as well for the field of obesity therapeutics. Next slide. I think even more reassuringly, we also see in the same studies a reduction in all-cause mortality. Now, why is that important? People with diabetes are at risk for many other diseases, particularly cancer. There are other issues that might be going on. And I think it's very important to show that you're not causing other harm while you're reducing cardiovascular benefit, while you're reducing cardiovascular events. And that's what we see with the GLP-1 receptor agonists. So how does this happen in humans? Next slide. And I have to say, this is more difficult to figure out. Mouse and rat have mainly an atrial GLP-1 receptor predominance. But when we went to look in the human heart and looked at biobanked heart biopsies, you can see all 4 chambers of the human heart, including the LV and the RV, have pretty robust GLP-1 receptor expression at levels maybe 5 to 10-fold lower than islet cells. So this is really a surprise to us and suggests that the actions of GLP-1 in the human heart might be very different than in rodents. Next slide. Even more interesting, I think, is the inter-individual differences in levels of GLP-1 receptor expression in these human heart chambers. And so there are up to 10-fold differences in the levels of GLP-1 receptor expression between one person and another. And is this, just a difference in sampling that the biopsies were taken from slightly different regions, or is this real biology? And does this perhaps reflect some degree of responder, non-responder? If you have more GLP-1 receptors, you're a better responder. So we don't know the answers to these questions, and it would be very interesting to find out. Next slide. So what about inflammation? And we know that if we say, what does obesity and diabetes and cardiovascular disease and liver disease have in common? It's clearly an increased inflammatory tone. But it's been very difficult to sort of where in the immune system the key GLP-1 receptors really are. So on the left, you can see we've looked extensively at macrophages. We don't see much. We've looked extensively in the spleen and the bone marrow and peripheral immune organs. We don't see much. But on the right, you see the major immune cell type that expresses the GLP-1 receptor. And that's in the gut. That's the intestinal intraepithelial lymphocyte. It's a canonical receptor. It responds with cyclic AMP formation. And the levels of GLP-1 receptor are pretty high, very similar to those in beta cells. Next slide. So why might there be this communication between the L cell and the local immune cells that express the GLP-1 receptor? So Fiona introduced this concept to you about these cells being chemosensors, and they really do sense a lot of bacterial, microbial metabolites and lipopolysaccharides and fermentation products. So we think there's tremendous dialogue between the contents of the distal gut, the microbiota, the L cells, and then feed forward to communicate with the local immune cells. But does this explain on the right these tremendous anti-inflammatory effects we see when we activate the GLP-1 receptor? We see suppression of inflammation in many organs very remote from the gut, where reduction of inflammation is quite profound. And I can tell you, we think the gut immune system contributes partially to some of these systemic and anti-inflammatory effects, but they're certainly not the whole story. Next slide. So let me, segue to GLP-2. We know the L cell is always making GLP-2 every time it makes GLP-1. And there were case reports, in the late 1970s and early 80s from the United Kingdom, from Professor Dowling and Steve Bloom, that if you had a rare type of glucagon-producing tumor, this produced bowel growth in humans. And so we recapitulated this observation in the left-hand panel, multiple different glucagon-producing tumors in nude mice, robustly stimulated gut growth. And it was really the simplest of experiments that you're seeing on the right, addition of multiple different peptides and assessing bowel growth. And glycine is a bit of a bowel growth factor, but you can see that GLP-2 is the more robust intestinal growth factor. And this is what I call the simple Mettler balance experiment, simply administering the peptides, taking out the gut, and weighing them. And what consequences did that have for clinical medicine? You can see in the next slide. And this really took 15 years or so. Jeff Flyer referred to the ups and downs and pitfalls of GLP-1 therapeutic development. This story was no different. But if you have intestinal failure and you're dependent on parenteral nutrition, you really can have a pretty good response with GLP-2 administration. You can reduce your number of nights on TPN, and most reassuringly, 15 to 20% of people in the clinical trials, and probably even more in the outpatient use of GLP-2, can completely come off parenteral nutrition. And this is a huge, I think, improvement in the quality of life for these individuals. Next slide. So we're still learning about what GLP-2 does. You know, one of the surprises to us was that GLP-2 markedly regulated gallbladder emptying, and Jens Holst and Philippe Knopp have recently shown the same thing in humans. And if one looks at the human liver on the bottom left, there's actually more GLP-2 receptor in the human liver, much more than, for example, the GLP-1 receptor. So not only is this gallbladder GLP-2 axis something to note in terms of side effects of GLP-2, but there's also some recent biology for GLP-2 receptor activity in the liver. Next slide. So if one looks for GLP-1 receptor expression in the liver, as I've noted, there's very, very little. And, you know, this is puzzling to us because, you know, GLP-1 appears to be in promising clinical development for NASH. And is this all weight loss and indirect reduction of inflammation? Or does this very faint GLP-1 receptor expression in the liver have anything to do with the hepatic benefits? But there's more GLP-2 receptor in the liver. And the bottom left, if you knock out the GLP-2 receptor and then stress the animals with a high-fat diet, you see profound inflammation. And what you actually see are stellate cell markers. Now, the top right shows there's no GLP-1 receptor in hepatocytes, no functional cyclic AMP response, and there's no GLP-2 receptor in hepatocytes either. But where there is the GLP-2 receptor is in hepatic stellate cells. And next slide. You know, this is very nice recent work done by Shai Fuchs in the lab with Bernardo Justa. And so it turns out that GLP-2 communicates not just with the gallbladder, but also with stellate cells to suppress inflammatory tone. And when you lose that signal, you have markedly increased stellate cell activation and increased hepatic inflammation in the absence of GLP-2 receptor signaling. So yet a new chapter of GLP-2 for us to try and figure out the importance of. Next slide. So coming to the end, I think it's important to recognize that this has been a very long journey that all of us have shared together, and many different individuals have contributed to this story over the last 35 years. And I have to say, it's been tremendously exciting to witness all of these various seminal events. Next slide. So although, you know, Joel and Jens and myself were fortunate to be there at the very beginning, and we're still delighted to be here, there have been so many colleagues that have made amazing contributions. You know, Steve Bloom with his first discovery that GLP-1 would inhibit food intake in rats, Werner Creutzfeldt on the left, really the pioneer of incretin biology. And it has taken hundreds of clinical investigators, many talented people in the pharmaceutical industry, and finally, so many, I think, generous people with diabetes and obesity who have volunteered their time to participate in the clinical trials that ultimately has enabled us to bring this story full circle from bench to bedside. And I'd like to thank all of these people for their many outstanding contributions to the story. Next slide. And certainly the current people in my lab and the many dozens of trainees who have really supported the science and done the hard work. I owe you a tremendous amount of gratitude, as well as the funding agencies. Thanks very much.
Okay, thank you, Dan, for just a spectacular talk. We'll just have time for a couple of questions. I do want to say first that thank you for recognizing the contributions of John Eng in his early therapeutic efforts with Exendin that are really quite notable. Also for the fact that so few companies were interested in pursuing his initial observations there. But a couple of people have asked questions that I'll ask you. So do you think, given what is now observed from your work and others about GLP-1 agonists and the cardioprotective effect and the anti-inflammatory effects. Do you see a point, or is it now happening, that these agents are being used independent of diabetes or obesity for their cardioprotective and or anti-inflammatory effects?
Yeah, so that's a great question. So we don't have approval in any country for that use, nor do we have the clinical trial data to support it. But I think it's very clear that these are not glucose-dependent effects. They almost certainly, I believe, would be demonstrated in people without diabetes. We have a great obesity trial underway with semaglutide that will be very informative. And I think, you know, companies should see this as an opportunity, as you point out, to really explore now in much greater detail the use of these agents in people without diabetes or without obesity, but with you know, difficult to treat atherosclerosis and cardiovascular disease. So I think that's a promising area for ongoing investigation.
And then along those lines, just briefly, do you think, would you anticipate any adverse consequences from the effects on beta cells, or would the glucose dependence of that process mean that it shouldn't cause any problems?
Yeah, so this is the beauty of the GLP-1 system. You know, the first exenatide approval was April 28th, 2005. So we have a 15-year clinical experience with the use of these agents. We were worried about pancreatitis. That's not an issue. We were worried about cancer. That's not an issue. Hypoglycemia is extremely rare, and weight loss is almost always beneficial. So I'm much more reassured now about the safety of these agents than I think we all three of us were perhaps 15 or 20 years ago.
Thank you very much. That is great. So at this point, I'd like to turn it back to Professor Khan for the cleanup part of the program here.
All right. Thanks, Jeff, and thank you, Dan, for a great talk. So we're coming to the fifth and final presentation, and it's my pleasure to be able to introduce a good friend and colleague, Matthias Chopp, to give this final lecture. Matthias is a physician, both a basic and clinical researcher, who is recognized worldwide for his contributions in the area of diabetes and obesity. Matthias received his MD from the Ludwig Maximilian University of Munich, and then went to do training in it, where he also went to do training in internal medicine. In 1999, he came to the US as a postdoctoral fellow with Richard DeMarchi at the Eli Lilly Research Laboratory. And it was really, I think, at this point that he became interested in the possibilities of novel peptide therapeutics that could be derived from existing peptides or combinations of existing peptides. In 2002, he returned to Europe to establish his own independent laboratory, but then was drawn back to the United States, to Cincinnati, where he came to work in their endocrine division and eventually became the director of their Diabetes and Obesity Center of Excellence and a professor of medicine. In 2011, Matthias returned to Germany, this time to Munich, to serve as scientific director of a newly formed Helmholtz Diabetes Center, and to convert this into a truly world-class diabetes center. Since 2018, he's become the CEO and scientific director of the Helmholtz Center, and is now responsible for leading this very large organization of over 2,700 scientists and staff who are focused on translational research discoveries. He is also chair of the Division of Metabolic Disease at the Technical University of Munich, and is the first German physician to receive the Alexander von Humboldt Professorship. He has an adjunct professorship at Yale University, and on a personal level, he's also a great drummer and has a fun band called the Sugar Daddies, which perform at a lot of meetings. But today he's going to tell us about his very exciting developments in the area of glucagon-like polyagonists, their discovery, their mechanism, their clinical translation, what I call the next generation of glucagon-like peptides. Matthias?
Thank you very much, Ron. Thank you very much, Jeff. Thank you for the invitation. Good afternoon or good evening, as it is here from Munich. I'd like to start by joining everybody in congratulating Joel and Jens and Dan to their achievements and the award of this special day today. certainly all the three of you have had huge impact on the lab's work here in Munich, on my career, and you continue to inspire us basically every day in the lab. Today, I've been asked to talk about the polyagonists, and next slide, please. I can only do so by first introducing Richard DiMarchi, my long-term collaborator. He was just mentioned. Richard and I have been working for almost 2 decades now together on trying to see if there's additional opportunities in maybe combining effects of several gut hormones into single molecules that have larger action profile and maybe more potent. And some of the successes of that work I'll share with you over the next 15 or 20 minutes. Next slide. So Richard, as a chemist, and myself as a ex-clinician turned physiologist, thought, well, if we want to fight obesity, we need to have some guiding hypothesis. And the ones that we put together said, well, we need to target the brain. It seems very clear that that has a major role, based on GWAS studies and based on some of our own experiments in the lab. We need to do this probably indirectly, and the gut-brain axis seems to be something that is working really well in that regard. We know from targeting endocannabinoid pathways in the brain directly that could be side effects easily. So using what we first seen in our lab with the effects of the gastric peptide ghrelin that turned out to be the hunger hormone, this axis between the gastrointestinal system and the brain could be advantages, but it was soon clear to us that it needs to be a polytherapy. It is not a single pathway, because there is clearly a pattern that the neuroendocrine system, the brain, sees and then reacts with the appropriate responses. Well, question, next slide, please, was which combination of which factor should that be? Because there's so many, and here's just a small selection of targets that are participating in this gut-brain talk. Now, the many mistrials and the many failed hypotheses we had, I will not share today. There isn't enough time for that. But what helped us is and it was mentioned earlier today, gastric bypass surgery success that I think is teaching us a lot of lessons, and it steered us in the direction of the GLP-1 glucagon system. Next slide. Because several of these peptides do respond and seem to return to more normal levels after gastric bypass surgery. Also, we knew that several of these players of the glucagon peptide family have receptors in these neuroendocrine systems that so powerfully steer our metabolism and energy balance. Now, looking at that family, it was, of course, Richard as a world-leading peptide chemist who saw that there are some similarities between these peptides from this family that can be used to build single molecules that could maybe activate several receptors at the same time. So dual agonists for that system seem to be entirely possible. And we wanted to start with GLP-1 combined with glucagon, which was deemed extremely crazy at the time, because glucagon clearly has effects on glucose metabolism that nobody thought were attractive. But glucagon also has effects in terms of energy balance and burning calories and maybe satiety even, that seemed interesting. So we thought, well, if the GLP-1 effects that we have been introduced to today in the last hour that are so beneficial for metabolism, that help lose body weight, can override the bad things that glucagon does, and then in combination, we get a powerful synergy that could be worth exploring. So in the next slide, you see data that have long been published now. Next slide, where indeed these single molecule dual agonists, GLP glucagon combinations, have shown first preclinically to have massive body weight effects. In obese mice, there was body weight loss that we could not achieve with anything else until then. In red, you see an unbalanced coagonist with about 10% glucagon action. In blue, you see about 100% glucagon action, always combined with 100% GLP-1 receptor activation. Now, of course, the big question was, and the prediction of many of our colleagues, something bad might happen with glucose metabolism. Next slide. Well, that didn't seem to be the case, because insulin seemed to be more or less normalized. Glucose tolerance improved massively. So indeed, the combination was advantageous, and GLP-1 seemed to balance out the potential diabetogenic liabilities that glucagon would bring into the picture here. Now, on the other hand, we didn't just want to have another GLP-1 and a super GLP effect. Therefore, we had to show that there is something else in these molecules. And actually, in collaboration with the previous speaker, Dan Drucker, next slide, please, we could show in mice that didn't have GLP-1 receptors that were functioning, because they were knockout mice for that there is still a decrease that is even larger when there's more glucagon dialed into the chemistry of the molecules that Richard was making in his lab. So there's a contribution of glucagon in these preclinical models to weight loss that is superior with that combination of these two factors. Now, next slide, please. Of course, we didn't want to treat mouse obesity. And now there are several papers out that show that this in principle works in humans as well. Several pharmaceutical companies are testing that concept. Here, two examples through the last 2 years. And next slide, there is some recent developments that suggest that specifically on fatty liver disease, steatohepatitis, NASH, there could be an even more important effect of these combinations of GLP, glucagon, dual agonists. There was recently a licensing deal between Merck and Hanmai that seems to be reflecting enthusiasm that in clinical trials, there is something possible with this dual agonist that cannot be achieved with monoagonism. Next slide. Still, a glucagon receptor activation in humans is something that needs to be tested thoroughly, and there's a lot of things we don't know. In parallel to this, therefore, we have been exploring other options. Next slide. As for example, and this was mentioned earlier today by Jens, a GLP-GIP coagonist, so-called twincretin. These are molecules that also Richard Slab was able to produce that were mostly in balance, activating both receptors. And we were predicting that this could be something where we really have an impact on improving glucose metabolism and making mostly a difference there. We didn't expect much effect on body weight because really the existing data at the point in the knockout models wouldn't have predicted that. Next slide. Well, turns out we were wrong, but wrong in an interesting way, because indeed these dual agonists for GIP and GLP receptors, they give us body weight loss that is better than what you get with monoagonism. Now, how could that be? Well, I'll share some unpublished mechanistic data in a minute, but first of all, we again wanted to see if this is relevant in clinical studies and in humans. Next slide. Turns out, next please, yeah. So it turns out this works in principle in humans. These were early studies with a collaboration with both Novo Nordisk and Roche that Richard and I were involved in. But more recently, and this was mentioned earlier today as well, next slide, Eli Lilly and Company showed with their compound tirzepatide that's based on this GIP-GLP-1 principle that we had published a couple years earlier, that there is a dose-dependent decrease in body weight that goes beyond what monoagonism here in the control group could achieve. And of course, next slide, this also is reflected in the impact here on HbA1c, glucose metabolism, and diabetes. Now, the reason this might work better, we always thought philosophically could have to do with the fact that there is receptor occupation throughout the human body that needs to be more to the limit and maximized with monoagonism. Because with dual agonism, with a lower receptor occupancy, you might get through the synergy still the same or better effects, and therefore have a larger therapeutic window and be less in danger in terms of side effects. And next slide, that might be the case because it seems when you look at the side effect profiles with dual agonism, GIP, GLP, that there is less than you would get with monoagonism when you would treat to the same benefits of what you see with the dual agonism. So maybe that is really where the advantage resides. We will certainly learn more now as these compounds are in phase 3 clinical studies and maybe getting close to approval. Next slide. Now, we wanted to know and still want to know how that works, and a lot of questions that are still open. So here's some recent unpublished data, again, from the lab, studies here led by Timo Müller in Munich in collaboration with Richard DiMarchi and his team, showing that there is a chronic effect when you give GIP, longer acting GIP, intracerebroventricularly in the brain that decreases body weight. So clearly GIP can accomplish body weight loss when you give it into the brain, of course, you know, the appropriate controls, etc. And, you know, for a while we thought there could be an effect that is contributed by downregulation of GIP receptors. That doesn't seem to be the case here, as you see on the right-hand side, where we observe that the GIP receptor expression stays the same. Next slide. Now, this is a pharmacology study that I just showed you that doesn't, you know, prove it yet, but this one is getting closer. What you see here is a mouse model that, again, Timo and their team made with collaboration of Christian Wolfram and others, knocking out the GIP receptor specifically in the brain, and then giving the dual agonist GLP-GIP, and indeed you lose the superior benefits of the dual agonist over the monoagonist in this trial in the knockout mice, which, you know, indicates that likely, yes, GIP does something in addition to GLP. This is mediated by the GIP receptor, and it's mediated specifically by the GIP receptor population in the brain. Next slide. Well, you know, we'll continue to study this, and there's an ongoing trial here with numerous experiments in vitro showing that, I want to summarize it in the way that there is no indication that what's going on a cellular level, a contribution by GIP just makes a better GLP-1 receptor effect. If anything, it's the opposite. So next slide. Things we see in terms of G-alpha-S recruitment, internalization, recycling of the GLP-1 or the GIP receptor, all point to the fact that indeed it's the GIP addition that gives additional benefits, and it's not just enhancement of GLP-1 action, also on a cellular level. Next slide. Now, you know, if you can do two of these GLP, GIP, and you can do a dual agonist with glucagon, GLP, of course, we wanted to do a triple agonist, and many of you will know that Richard succeeded with that in the lab as well, and made a triagonist for the GIP, GLP, and glucagon receptors. Next slide. This triagonist, next slide, has been published already with its sequence that you see here. Just, you know, to go one more time over the fact that this isn't just sticking several gut hormones together to make a larger molecule. This is really engineering every part of a relatively natural looking gut hormone so that it becomes a polyagonist and hopefully has low antigenicity, because not a lot of this is changed in terms of what the body would see anyway in terms of its own gut hormones. Of course, you want to protect against DPP4 cleavage. You want it to be albumin binding, and you want to have a CX tail for pharmacological reasons. But still, this is very close to what's known from the monoagonist molecules that are currently in patients and approved. Next slide. So the triagonist, even if you compare it to a dual agonist like tirzepatide or similar GLP-GIP dual agonists, as you see here in red, in blue, the triagonist is even better. The body weight effect, the blood glucose normalization, the fat mass effect, and again, the effect on fatty liver in these preclinical studies we published here in Nature Medicine a while ago is really impressive. Now, next slide. That communicates to the fact that, again, Hanmai, the company I was mentioning earlier, has another therapeutic against NASH, which is a triple agonist like this one, glucagon, GIP, GLP. So it really communicates with our published findings that this triagonist works particularly well cleaning out the fat of the liver. And I'm very curious now, since this has been fast-tracked by the FDA, to see what will come out here clinically. Now, the next slide, please. This leads to this current landscape, thanks to Brian Finnan, who shared the updated version of this with me recently. It's a very busy field, very complex, always new players and some players that leave the field, but a lot of interest and a lot of ongoing clinical trials for all three classes: dual agonists, GLP-glucagon, dual agonists, GLP-GIP, and the triple agonists, which are also still being tested clinically. Now, I'll close with a little bit of a teaser into the future. Next slide. Over the last couple of years, we have been working hard, making a unique sort of a Guinness Book of Record mouse, a triple receptor knockout mouse that doesn't have GIP receptors, doesn't have glucagon receptors, and doesn't have GLP-1 receptors. So the CRISPR technology nowadays, next slide, makes that possible and allows us to really now have a littermate controlled cohort of the triple receptor knockouts, but also have all the dual receptor knockouts and the monoreceptor knockouts, etc. So that's a huge undertaking. 16 groups of mice that now under different conditions are being phenotyped for metabolism. Next slide. And what I can tell you so far, it's early days, but they're alive. They look pretty normal. They have normal blood glucose. And of course, it will be very interesting to see how these mice, the triple receptor knockout, respond or don't respond to several of the compounds that is shared with you or some other compounds. How important is the system in response to outside stimulation or challenge by pharmacology? So with that, I'll end and have to thank my collaborators. Next slide. All over the world. This is just a small selection, but I want to mention again Timo Müller, who runs the lab on a daily basis, together with Aaron and Alberto and Kian, and Richard DiMarchi, without whom that would never have started or be possible, Brian Finnan, who's now a director at Novo Nordisk, and collaborators. Dan is one of them, Randy Seeley, Christian Wolfram, Davide Calibero, and many others. I'm sure many that I have not even on this slide today. Next slide, please. This requires lots of funding. Alexander von Humboldt Foundation was mentioned, DRC needs to be mentioned, and many collaborators, but also the Helmholtz Association. And finally, I'm supposed to show this slide for the personal conflict of interest statement. Thank you very much for the invitation and happy to answer some questions.
Okay, well, thank you, Matthias, for a great lecture. As you know, we're running a couple of minutes late, but I do want to give you a couple of questions before we close the symposium. And we got a couple of questions here about how do you explain improved metabolic control with a GIP receptor antagonist by an antibody when combined with a GLP-1 receptor agonist? Is something missing?
I'm afraid I can't entirely explain that, but I think it's probably a different thing if you activate that system with a peptide or you inactivate the system with an antibody. Also, it is a tricky thing to find out in vivo and in different species if in the end what the cell sees sums up as agonism or antagonism. So, without further stirring the controversy, there's some mystery left to be cleared up.
Okay, and maybe one last question, which is, could you distinguish whether these dual or triagonists are truly synergistic or as opposed to additive?
Yeah, we have certainly seen that they go beyond just being additive. So over the last 15 years, numerous experiments showed that even if you go, sub-threshold, or if you, with the right comparisons and controls, test for additive phenomena, it does go beyond that. So I'd say that for most of the compounds I've just shared with you, it's true synergism. True,
terrific. Well, I'm going to close this part and turn this over to George. But before I do, there was a question that came in. Will the slides be available to others after the symposium? And I do understand that this symposium is being recorded and will probably next week be posted on the Warren Alpert website. So for all of you who are listening, you can both look again at some of the data and share it with your colleagues. And now I'll turn this over to George for his closing comments. Dr. Daly.
Great. Well, Ron and Jeff, thanks for moderating, and thanks to the speakers for such outstanding talks. What an exciting area of science. And thanks to all of you who participated today. I want to just quickly close and say we certainly hope that in the not too distant future, we will gather for next year's annual Warren Alpert Foundation Prize. And maybe it will be on something like solving the COVID-19 crisis. No matter what, it will be remarkable science as we saw today. Thanks to all, and be well and stay safe.